Hi, measuring and monitoring of biodiversity is essential for conservation practices. Before we begin any bioconservation activities, we should know how much biodiversity we are having. In this session, I am going to talk about how inventorying and monitoring of biodiversity is done. Biodiversity inventorying and monitoring is basically the identifying the elements of biological diversity and monitoring their changes. The elements of biodiversity including the genetic elements, species element and ecological elements. The genetic diversity is the diversity between the individuals of same species. And the species diversity is the number of different types of species we are having in an ecosystem. And finally, we have the ecosystem diversity. That's how much different communities and we are having in a particular ecosystem. In inventorying, what we are doing, we are identifying these resources, how much different types of resources we are having in a particular ecosystem. And in monitoring, what we are doing is like we are measuring the changes, how it is changing, the type of individuals are changing over time. And in order to get a proper inventory, all species and ecosystem need to be identified, named, cataloged, and studied. So it including most of the biotic and abiotic factors. So all of them should be identified, named. In the case of organism, we should have a scientific name that implicate the evolutionary origin of the organism and should be catalogued, and its ecological roles and functions should be studied. Then only it will make a good inventory for the biodiversity. And once we have the inventory, it will help us to determine how well various ecosystems are performing. As we know, the performance of ecosystem is directly proportional to the biodiversity. So, in order to system should be performing, it should have biodiversity. So, if you want to measure the performance of an ecosystem, we should know how much biodiversity we are having, and we can compare this biodiversity performance or the ecosystem performance for various uses, maybe for the conservation efforts. If we are running some conservation programs or we are protecting the ecosystem, uh, in order to know whether the efforts for protecting the ecosystem is useful, we should know how the biodiversity is changing. So, or if you are if you are planning, if the government is planning some projects which including some level of destruction of the environment. So in order to measure how much biodiversity or the ecosystem function we are going to lose, we should know how much biodiversity we are going to lose. So for so many purposes, we use the biodiversity inventory and monitoring. The inventory and monitoring of biodiversity is so complex. There are different reasons for that. One reason, or the main reason, is Earth's biodiversity is largely unknown. And the actually out of this whole diversity, what we know is about 2 million. If I say exactly, it is 1.4 million species are actually named. And few species we already know, but we are don't have the systematic classification of the organism. So roughly around we know 2 million of species in this world. And we don't know how much species is there. Estimate says that we at least will have 10 to 100 million species in this earth. So we know about maybe 1 to 10% of biodiversity. And here I am talking about cataloging or inventorying the biodiversity. So even if we classify, make a list of all this organism, it's only 1 to 10% of total diversity. Again, the 99 to 90 percentage diversity is missing. So we have huge number of organisms, and out of this large number, we know only a small portion. So it's very difficult to make a complete inventory. And I could say the making a complete inventory is more or less impossible. Next thing is that information on biological richness is mainly based on vertebrates and vascular plants. As I said, we have classified about 1.4 million organisms. Out of this organism, vast majority are plants, I mean large plants and animals. The diversity of the maybe insects, nematoids, bacteria, virus, fungus, everything is missing, largely missing. So we know only a small proportion of the diversity of a small proportion of the groups. It is easy to identify or find these bigger plants and animals. But when it comes to the viruses, it are too small. Unless otherwise they are making some problems like COVID-19 or some smallpox disease or the other type of diseases, then only we will know that type of organism is actually existing. Otherwise we won't know. In the case of bacteria, same thing is there. We know about the most of the pathogenic bacteria, the bacteria which is not pathogenic, or maybe in cases helpful bacteria, or bacteria which doesn't have any effect or any health benefit or health adverse effect. We don't really know these organisms exist or not. So our knowledge of biodiversity is largely unknown, and the most of the organisms are not not classified or not named. So that's one difficulty in inventorying and monitoring of biodiversity. But even though we know only a small percentage of biodiversity, by measuring or monitoring changes in this small percent of biodiversity, we can actually extrapolate the effect of whole biodiversity. That's the point. Even though we don't know how much biodiversity we have, but from the non-biodiversity, we can infer what is the possible change in the unknown biodiversity is having. So that's what I'm going to talk about in following sessions. So first I would like to distinguish between the biodiversity inventory and monitoring. Inventory is just like a stock take, like just what we do in the laboratory. When you first come to the laboratory, most of the people ask you to take a stock, how much chemicals you have, or maybe some of the glasswares will be allotted to you, and the teachers say you should have a list of glasswares you are having right now. That's just a like stock list. That's actually inventories. In the bio inventory, what we do, the status of a resource at a point of time. Like in a particular point of time, the inventory says how much biodiversity we have and how much abundance we have. This biodiversity. That's what the inventory is. It's just a list of organisms, a list of uh, resources actually. If we have a forest, the inventory of the forest says that what all resources we have, what all the biological resources we have, what are the non-biological abiotic resources we have in that forest. And it is a distribution and composition of species, habitat and population. It does not only include the number of individuals or number of species, it also say the habitat. So it's a mix of species, habitat as well as the population. So it's a list. It is made only once in a way. Monitoring is somewhat similar to inventory. Monitoring is actually inventory changes with the time. So when you come to the lab, some of the glasswares were allotted to you. After 3 years, when you are going after the PG or UG, whatever it may be, uh, you will take another list. You will compare the glasswares you have with you and with the list. Maybe you have given maybe 100, maybe 10 conical flasks. After 3 years, we are looking how much conical flask you are having. So maybe the few of them broken. Now you can only find seven. So 10 were given. Now you have only seven. That's actually the monitoring. You are after 3 years, you are monitoring how much biodiversity we have. So it's actually the monitoring is actually inventory change with time. In the case of species, in a forest, you are counting the number of tigers. In the 1 year, you count you have this much number of tigers. The following years, you are again counting. Every year, you are counting number of tigers in the forest. You can actually get the trend whether the number of tigers is decreasing or the number of tigers is increasing, or you can actually make conclusions based on the uh, trend of the population size of the tiger. That's what the monitoring do. So inventory is a one step process, or the it is done only once. You just take the list of everything, but monitoring is actually looking at the how the inventory is changing, how the uh, list of the biodiversity or the biological resources are changing over time. It's actually monitoring is actually the resource dynamics, how it is changing, whether it is increasing or basically it is the dynamics. From this monitoring, we are inferring the change or trend over time in the resources, in the terms of resource abundance, and the condition of the resource. And monitoring will tell you what the rate of change is, whether it is in what rate it is increasing or decreasing. If you are looking at a population of a tigers, how fast it is decreasing or how fast it is increasing, what's the rate? From the rate you can actually predict what will be the number of tigers after 10 years. So you can actually make future predictions based on the rate of change. And it will also gives clues for the causes of change, whether it is because of deforestation. In 1 year you have a huge reduction in the number of particular organism, particular species. You can actually correlate this decrease in number with some natural calamities or the human man-made activities, or in other time maybe you will get a good rise in number of species. You can actually correlate this changes in the number of species or the biodiversity with the local events, or some cases the global events also. And so the monitoring will tell you what rate change is happening and what is the causes of changes. Then it will largely affect the conservation practices. In the monitoring or inventory, the many times we are doing or the government is doing the process to know that how much biodiversity we have in a particular area. Suppose government plan to build a water reservoir inside a forest. Maybe they have two different places, potential places to making the water reservoir. So in order to know how much biodiversity or how much harm it is going to cause, we should know what is the biodiversity in it. So if we have a inventory, or we have monitored the changes in that particular area, we can actually say out of these two different potential sites, which is more harmful for the environment and which is less harmful. So, or maybe some conservation practices, like maybe the sometimes the forest area need to be increased. So that type of things should be done based on the number of species. If the in Kerala itself, one time what happens, the number of tigers increases. So what the government do, they take the some of the estate and incorporate it with the forest, existing forest, so that the population will flourish more. And sometimes the decisions, like nowadays the number of elephants is increasing very in high rate. So we should do something, like maybe we should increase the forest area or decrease the number of species. Then only the carrying capacity of the forest will be maintained. So that those type of decision will be taken by monitoring the change in biodiversity. So I guess you understand the difference between inventory and monitoring. Inventory is just a stock taking. It is a one-time event. We are just taking the list of all type of possible resources, bioresources, in a particular area. In monitoring, we are actually measuring the change, how this inventory or the how the biodiversity is changing over time. So first I'm going to discuss more about the inventory. Inventory is the state or status of one or more resources. The state or status can be presence or absence of a particular resource, or the abundance of a resource, density, or distribution. The in case of if you are looking for a looking for a animal which is under threatened, it is in the red list. If it is in the red list, we should know whether the particular animal is present in a particular ecosystem or a particular geographical area. So we can say the presence or absence of a species. Maybe if we are looking at the exotic species, we can say whether it is present or absent in a particular area. Its abundance is important, how much number it is seen, or density, how thickly it is populated, or the distribution, how it is distributed over the landscape. And inventory basically gives both a quantitative and a qualitative description of the bioresources. Uh, and basically, an inventory consists of three different things. Firstly, legal conservation status of the national estate. Most of the inventories are made at national level. Different nations will make inventories in their own way and in their own geographical area. Some of the nations are having more complete inventory than others. So, first thing to have is the legal conservation status of national estate, which is the conserved areas for biodiversity, and how much area it is having, what are the locations, what are the diversities having, what are the different landscape there it is having, so things like that. Secondly, extent of various vegetation and landform types within the geographical area. Finally, variety and abundance of biota. That's what we are interested in. Variety and abundance of biota. That's what the biodiversity stands for. So, in the following discussion, we are going to discuss only about the portion of inventory which consists of variety and abundance of biota, not about other things. So, bioinventory, it is actually measuring the biodiversity in a particular area, typically confined within a specific area. It may be a forest, it may be a aquatic, or maybe a portion of ocean, it may be a river, whatever it may be, it will be confined within a specific area. So the measure is for a specific area. The size of the area may vary. It may be a huge area or huge landscape, or it is a small area. That everything can be measured in the bioinventory. It determines number and relative abundance of the species. So how much number of individuals are there, and what is the abundance related? Relative abundance is the abundance of a species in relation with the other species. If you are looking for the herbivorous organisms, like we are measuring the abundance in a particular trophic level. We can measure the abundance in the producers, maybe in the primary consumers or secondary consumers or predators. In the same trophic level, how much abundant it is. That's what the relative abundance say. So bioinventory contain number of organism and relative abundance, and it is created at appropriate spatial scale and for appropriate duration. The spatial scale of a inventory is like, in sometimes the we may be looking for a common species. Making an inventory of a common species is easy. It is easy to find. Sampling is easy. If you look around, could find those type of species very easily. If you are making an inventory of some type of, like inventory of a common bird, it is easy to find that those birds. But if you are making an inventory of rare birds, it's very difficult to find. And if you have only few number of samples, the chance of error is high. So making an inventory for a common species is easy, but making an inventory for rare species is always difficult. And the duration of the study is also important. Sometimes the duration for sampling and the creating the inventory, we are actually making a list. So in order to make this list, if you are looking for the migratory birds, the birds will come only to the particular geographical area maybe 1 month in a year. So in order to make the inventory of those species, you need to spend only 1 month. But instead, if you are studying the life cycle of a organism, the whole life cycle of our organism, which lives for 10 years, you may need to spend more time for that. You need to spend the duration of study will be high. So depending on the objective and the type of organism and the type of study, the duration of study may vary. Assess the presence or infer the absence of species within an area. In an inventory, we need to assess the presence, or how much number it is seeing, whether it is present in abundance or the high density, or it is absent. It is especially important when we are looking for conservation efforts. Some of the organisms like panda, they are very few in number. It's very difficult to find them in forest. So assuring its presence or absence is very important. For any inventory, finding the presence of our organism is easy. Like if you see one organism, you can say you have this much number of organisms. But confirming the absence of organisms is very difficult. We cannot be sure, 100% sure, that in this forest of this much hectare area is not having the particular organism. Maybe it will be there. Especially it is very difficult for confirming the absence of an insect. Insect is too small. So maybe you are sampling, you may miss the insect, or it may have changed the habitat, or it may have changed the location. So for an inventory, it is easy to find the presence of organism, but it is very difficult to report an organism is absent in a particular area. And bioinventory aid resource planning and species conservation. Resource planning is like just what I say, if we are planning some projects or the cultivation or deforestation or something like that. we should have a good idea how much biodiversity we already have, so we could actually measure the level of damage we are going to cause to the ecosystem. Otherwise we don't have any idea what's going to happen. And the species conservation efforts, the bioinventory will give you an idea how effective our strategies are, the preservation or conservation efforts are, how successful our conservation efforts are. So that's important. These are the different steps involved in making a inventory. So the steps begins with the setting the goals. The inventories are made with the different intentions. Sometimes it is just reporting the number of individuals. Sometimes we are more interested in conservation. Sometimes we just want to measure how much uh, impact a project will cause. So things like that is there. So the setting the goals. So what are the objectives of inventory is the first step. Secondly, the identification of resources to in inventory. What are the resources? What are the biotic and abiotic resources we should want to put in the inventory? Maybe the parameters like more sensitive to change used to be in the inventory, like abundance of a population or the density of a population or the breeding pattern. Things like that can be involved in a inventory. The resources which is selected for inventory should have a direct relationship between the biodiversity. Then we establish the trigger point. Trigger point is the set value or the like benchmark. Before starting the study, we are setting the benchmark. If I find this much number of tigers in this forest, I could say the tiger population is good. Population is like very good in this forest. Its number is enough. Things like that. So we are fixing the benchmark before the study. So how much, what is the expected value, or we are fixing the benchmark in some points, the some numbers or something like that. So if you our after the study, if we found more number of organisms in this particular area, we will do that. If we found number below that one, we will do another, will go other thing. Or maybe if you want to make a reservoir, a water reservoir inside a forest, in the inventory we can say if you see biodiversity above some point, we should not build that because this very diverse population. Or if you see number of biodiversity below that point, we can say we could make the reservoir. So decision making points or the benchmark can be set before the study. Then developing sampling design, which is very important, because for a large geographical area, it is very difficult to in go and study all possible locations. So that's difficult. So what we do, we will take samples from different locations, and using some statistical techniques, we will combine the data and extrapolate to the whole region. So sampling design, sampling technique, sampling area, sampling time, everything is important. Depending on the type of organism, maybe we'll go for the sampling at night, or we will make some traps for the organism, or we may fix a camera. So many strategies will be applied for sampling. Then once we have fixed the sampling design, we'll collect data uh, using different techniques. Then go for the analysis. Analysis is mostly done by computers using the statistical softwares. So the whole data will be processed and analyzed. And once we have the analysis done, we will go for the evaluation of result, and we will compare our result with the benchmark. So we'll have a good idea of what is going on, whether it is improving, whether it is good or bad, something things like that. Once we have the result, it will be used for decision making. Sometimes the government decisions or legislations or the multinational agreements, things like that will happen based on the uh, this abundance, or this result of this study. Some spaces are declared as the biodiversity hotspots based on the inventory, how much biodiversity it has in a square kilometer, how many different types of organisms you find. So those all come from the inventory. So that's all for this session. Thank you so much for listening. Thank you.